This is ANAplus, powered by the African News Agency. My name is Lance Witten. You're watching Newsbite, where we give you everything you need to know about Africa now. It was recently announced that researchers had found a way to cure mice of HIV infection. But the exact origin of the virus HIV-1 has been a matter of debate ever since its discovery. There's now a wealth of evidence on how, when, and where HIV first began to cause illness in humans. In 1999, researchers found a strain of simian immunodeficiency virus called CIV-CPZ in a chimpanzee that was almost identical to HIV found in humans. Now, the researchers concluded that proved chimpanzees must have been the source of HIV-1 and that the virus had at some point crossed species from chimps to humans. The same scientists then conducted more research into how SIV could have developed in the chimps. They discovered that the chimps had hunted and eaten two smaller species of monkey. These smaller monkeys infected the chimps with two different strains of SIV. The two different SIV strains then joined together to form a third virus, CIV-CPZ, that could be passed on to other chimps. This became the strain that could also infect humans. But how did HIV make the jump from chimps to humans? Well, the most commonly accepted theory is that of the hunter. In this scenario, CIV-CPZ was transferred to humans as a result of chimps being killed and eaten, or their blood getting into cuts or wounds of people during the course of hunting them. HIV is a type of lentivirus, which means it attacks the immune system. In a similar way, the SIV attacks the immune systems of monkeys and apes. Since its first identification, almost 3 decades ago, the pandemic form of HIV has infected at least 60 million people and caused more than 25 million deaths. But as of last week, there seems to be light at the end of this dark tunnel. Researchers have successfully eliminated HIV from the DNA of infected mice, a promising step towards a cure for the millions of people living with the virus. The researchers tested their methods on a group of infected humanized mice. Those were rodents engineered to produce T cells similar to those found in humans, which are susceptible to HIV. Combining CRISPR, a tool that can edit genes, with long-acting, slow, effective release antiretroviral therapy, a recently developed therapeutic strategy, the researchers were able to effectively eliminate replication-competent HIV DNA from the genomes of approximately 30% of the infected humanized mice. By the study's end, researchers had successfully eliminated the virus from 9 out of 23 mice. The results prove that HIV can be eliminated, the researchers said. Stephen Deeks is a professor at the University of California in San Francisco who has worked extensively on HIV. He said the use of gene editing to remove HIV from a live animal is a notable step forward. But he cautioned that using the technique on humans would be far more challenging. Scientists would have to grapple with more variations of the virus, more difficulties in delivering the gene editing technology, and the possibility of cutting up human genes while trying to target HIV, he said. Those are formidable problems, Deeks said, especially with success depending on removing the virus completely. While there is clearly a long way to go, it is a step in the right direction. Now, why is this important for Africa? Well, with under 20% of the world's population, Africa is home to more than 60% of people living with HIV. South Africa and Botswana have the highest infection rates in Africa, but countries in Africa have also been on the forefront of adopting interventions and treatments to tackle the pandemic. Well, this is Newsbite on ANAplus. We make Africa matter.